to tell the sequence by binding to the outside, but to actually turn the DNA into a protein, you've got to be able to unzip the molecule. And since the bonds that hold it together are relatively weak, it's easy for the cell to unzip the double helix without heating it up, because cells can't tolerate heat. They use enzymes that can ratchet these. If these are covalently bonded together, you can never unzip the code and read it to make mRNA or protein. But having weak hydrogen bonds here is relatively easy for the cell to unzip it, read the code, replicate it, transcribe it in RNA, and then put it back together. Now in the laboratory, when you make DNA go single-stranded, we call that denature the DNA, or melt it. We do that in the laboratory with heat. When you heat DNA, you don't want to boil it because you break these covalent bonds and fragment the DNA into pieces. But if you heat DNA up, it'll simply shake apart. These hydrogen bonds are too weak to hold together. They'll shake it apart. You can then clone it in a test tube. Or you can hybridize it to another DNA molecule to see how similar it is to some other. You take maybe DNA from a chimpanzee, from a human, melt it into single strands, let it cool, and see if they'll hybridize together if they have a similar sequence. And they do. They hybridize beautifully because we're so closely related to chimpanzee. Do with us in a fruit fly doesn't match up so well, doesn't tend to hybridize as well. Okay. So this structure is well suited for cells to be able to read the code and not heat themselves up, and it's well suited for molecular biologists to be able to melt the DNA and manipulate it in the laboratory. We're going to do that in what's called the polymerase chain reaction next week. We're going to clone DNA in a test tube, and then we're going to cut restriction enzymes to see what version of a gene you have. Cool lab. Now let's get back. So we understand the nature. Remember the two goals of determining this three-dimensional structure. One was to determine how the genetic information is stored in the DNA, right? It's the spelling of nucleotides along one strand. Okay? What about how does this structure help the cell to replicate DNA prior to cell division? Well, the cell can separate the two strands of the parental molecule. This would happen in the S phase of interphase. And then each parental strand in gray becomes a template to which nucleoside triphosphates, that's the monomer here, can be added according to base pair rules. You end up with two new sister chromatids in the original chromosome. And notice, and what we understand to be the correct mechanism of replication is that the new sister chromatids are half old, half new. And their sequences will be exactly the same as the parent, right? Because this template serves to bring in what's called the complementary base pair. Does that make sense? So in the simplest version here, if I were to draw just like a straight ladder, just so you know what I mean, good hands here. So imagine about a short stretch of DNA here. I just put a few bases here, C, T, G, and A. I'll use all four there. Of course, the complement would be G, A, and C, and T. I want to replicate this. We just separate the two strands. In the cell, enzymes do this. In the laboratory, we do it with heat. Separate the two. Now the parental strand becomes a template. There's the C, T, G, A. Of this parental strand, over here it's the G, A, C, T. And then the cell imports from the nucleus, or in the test tube, we add it to our reaction mixture. We add G's and C's and T's and A's, and of course, enzymes attach these, G to C, A to T, C, T. Because remember, only the complement nucleotide can hydrogen bond correctly. And you see at the end of replication, we've got two molecules that have the exact same sequence as each other in the parent that no longer exist. So it's the complementary base pairs proposed by Watson and Crick that explain how replication can occur, how daughter cells and descendants can carry the same genetic code into the future. Now this mechanism of replication, where the parental strand separates and each strand becomes a template, the addition of new monomers, this is called the semi-conservative model of replication. Semi-conservative model. Now, when Watson and Crick proposed this, they weren't sure this is how it worked. It was a hypothesis. Okay. They did no original chemistry themselves. It was all using other people's information and lots of good ale and cocktail napkins. That's how I get a Nobel Prize sitting in a bar, writing ideas on cocktail napkins. Works for me. So how do we know this mechanism is correct? Well, the experiment was performed in 2 years after Watson and Crick's paper in 55 by Messels and Stahl. Your book has a nice page about this experiment. Let's look at it briefly. There were 3 possible models of replication that Watson and Crick didn't distinguish between because they didn't have any data to know. Here's the semi-conservative model that turned out to be correct, where you can see the parental strands are gray, and they separate, again, by enzymes in the cell, by heat in the test tube, and then monomers are added according to base pair rules, and each new daughter strand is half. So after 1 generation, they're half old, half new. And after 2 generations, well, now we've got some molecules that are, this is the old one here, but you can see, the original parental strands only last a generation, right? Okay. Well, they still exist, but basically they get sort of diluted out. It was possible that there was some sort of conservative replication where the parental molecule remains intact and its sister chromatid is made out of all new monomers. And so by the 2nd generation, the organization is like that. It gets more and more diluted out. And then there was this dispersive model where perhaps maybe the parental strands are broken into fragments, and you get like a patchwork, a quilt work. Okay? So there are 3 possible models of replication. So Messelson and Stahl developed a method to distinguish between these 3 methods. Okay, let's look at the summary of a very brief overview of what they did. And this is in your text in case you don't quite understand this. What they did is they grew bacteria in a flask with nitrogen 15. Now, nitrogen 15 is the heavy isotope of nitrogen. The normal isotope of nitrogen is nitrogen 14. It's got 7 protons, 7 neutrons, 7 electrons. This has an extra neutron. So 7 protons, 7 electrons, otherwise it's not nitrogen, and 8 neutrons. So it's heavy. Ooh, it's heavy, man. Groovy. Okay? All right? So they would grow bacteria, okay, from scratch with this heavy nitrogen, okay? And then they would transfer the bacteria to the normal isotope. And after 1 replication, they would extract the DNA from those cells, the first generation of offspring after the transfer. Then they would extract DNA from the 2nd generation. So they grow the bacteria in heavy nitrogen, would become part of the nitrogenous bases. So they're called nitrogenous bases. You start them growing, and then you transfer a batch of these bacteria to a new culture media that has no heavy nitrogen. It's got the normal isotope. It's one atomic weight lighter. And you let them replicate for 30 minutes, let them divide 1 time, then extract DNA, then let them divide a 2nd time, extract the DNA. So you got to do this, like you transfer them, 30 minutes later you extract the DNA, then 30 minutes later you extract the DNA. Then you put the DNA extract into a column containing cesium chloride. That's the salt they were put in. Okay? This is a pretty heavy salt. And you spin it in a centrifuge at high speed. And if you spin things in a centrifuge, we know that heavy things go to the bottom, light things float. That's the idea here. So it's a tube of cesium chloride. That's what the yellow is here. Okay? And then they could detect the DNA. I'm not sure the detection method. I never read the paper, but I'm not sure if they were using the radioisotope detection methods or some other method to detect the DNA, but they had a detection method. And they looked for the band of DNA. And if you extract the DNA from the original culture, you had 1 band of DNA that sunk to this level because it was completely consists of the heavy nitrogen. After 1 replication, they saw a lighter band, 1 band that was lighter, a little less dense. And after the 2 replications, they saw that band, but then they saw a lighter band. Okay? So the band began to migrate up, and then this band persisted, but a new lighter band appeared. So how do they interpret that? Well, here shows you, we compare the three models again, so they're in your face. Here's the semi-conservative model, okay? And if you see at the beginning, the DNA is all heavy here. Then after one replication, you see the DNA is half old, so it's, here I am here, got to go this direction here. So here we are, the parental generation. After one replication, the band's moved up because the DNA is now kind of half as heavy as it was. And then after that, these still persist, so that band is still there, but now you got a lighter band here. Okay? Now, conservative was the explanation. This is what they saw. Okay? If it had been a conservative replication, this should have happened. You should have had, again, a heavy band. Okay? And then after the next replication, you'd have that heavy band and a light band. Okay? And then that would persist because you still got the heavy and the light band, right? If it was dispersive, you start off again with the heavy band, and then it just gets lighter and lighter. Just keeps getting lighter and lighter. So those three possibilities, I'm sorry, this is the one they saw. And so this confirmed that the method of replication is semi-conservative. Does that make sense? Okay? Very clever, folks. I think they're Americans, actually. Well, it doesn't matter. Make sense? Anybody have a question about that? Don't just it's going to get more complex as we keep going, don't you? It's going to keep ramping up, even this lecture today. You going to move forward or not? Yeah. So let's look at the so go ahead. Let me let you ask your question before I interrupt. Go ahead. So let's focus on the correct model, and the data are right here. So when they grow the bacteria at heavy nitrogen,